Greetings to all. Now we are going to see about tetracycline. And these are the contents of tetracycline: introduction of tetracycline, classification of tetracycline, structure of tetracycline, mechanism of action, mechanism of resistance, production, uses of tetracycline, and then storage of tetracycline. First, we are going to see about introduction of tetracycline. Tetracycline are broad spectrum antibiotics, which are chemical substance produced by a microorganism that are able to kill other microorganisms without being toxic to the person, animal, or plant. Tetracycline were derived from Streptomyces bacteria. Tetracycline were discovered as natural product by Benjamin Mayen Duger in 1945 and first prescribed in 1948. Then the classification of tetracycline. These are the duration of tetracycline. First duration, tetracycline, chlorotetracycline, oxytetracycline are acting in short acting 6 to 8 hours, and then demyclocycline, metacycline are intermediate acting in 12 hours, and doxycycline and minocycline are long acting in 16 to 18 hours. And then this is the structure of tetracycline. Mechanism of action in tetracycline. Tetracyclines enters stable organism by passive diffusion and by an energy dependent transport protein mechanism unique to the bacterial inner cytoplasmic membrane. Tetracycline concentrate intercellularly in stable organism. The drugs bind reversible to the 30th subunit of the bacterial ribosome. These actions prevent binding of tRNA to the mRNA ribosome complex, thereby inhibiting the bacterial protein synthesis. In this, the mechanism of action, tetracycline target the 30S ribosome, 30S subunit, and prevent the binding of tRNA to mRNA, and then it contain amphisilin tRNA, or it gives the binding of mRNA ribosome complex, thereby inhibiting the bacterial protein synthesis. And then mechanism of resistance. Cell become resistant to tetracycline. There are three types of mechanism of resistance. First, enzymatic arrest, second, influx resistance, and then ribosomal protection. In enzymatic arrest type of resistance, where an acetyl group is added to the molecule causing inactivation of drug, and then influx resistance, gene encodes a membrane protein that actively pumps tetracycline out of the cell. And then third one, ribosomal protection, which locking tetracycline from binding to the ribosome. And then now we are going to see about production of tetracycline. The tetracycline was produced through shaker flask fermentation of Streptomyces coleoclerc and Streptomyces aerophaciens. Production of chlorotetracycline. All media contained less than 1 ppm chloride. In production of tetracycline, the base synthetic medium which developed for the fermentation and has the following formulation per ml: sucrose 30 gram, trisodium citrate 2.5 gram, ammonium sulfate 3.3 gram, magnesium sulfate heptahydrate 0.25 gram, monopotassium phosphate 0.10 gram, and dipotassium phosphate 0.10 gram, calcium carbonate 1.00 gram, 1 gram, magnesium sulfate 0.1 gram, zinc sulfate 0.04 gram, potassium dichromate 0.016 milligram, and acetic acid 0.40 ml. The production of tetracycline, the fermentation process concerned with the production of cephalosporin, is similar to that of penicillin. The culture medium consists of corn steep liquor and soya flour based medium in a continuous feeding system. The other ingredients of the medium include sucrose, glucose, and ammonium salts, and then methionine is added as a source of sulfur in the production of tetracycline. Preparation of culture. Isolation or carried on waxman agar for the isolation of soil microorganism. In 1922, waxman had done without the adjustment of pH and were incubated at 30 degrees Celsius for the period of 10 days to permit good sporulation. The formula for the agar follows: glucose 10.0 gram, peptone 5.0 gram, mono potassium phosphate 1.0 gram, magnesium sulfate heptahydrate 0.5 gram, agar 20.0 gram. Selected isolates are stored under oil and are lyophilized. In the inoculum medium, sodium citrate 12.8 gram, sucrose 40.0 gram, ammonium sulfate 6.0 gram, magnesium sulfate heptahydrate 0.25 gram, mono potassium phosphate 0.15 gram, calcium carbonate 11 gram, magnesium sulfate 0.01 gram, zinc sulfate 0.044 gram, potassium dichromate 0.016 milligram, or the inoculum medium. The inoculum preparation: volume 50 ml culture per 250 ml medium, 400 ml per 2000 ml. Shaker stock at 97 cycles per minute. Temperature is 30 degrees Celsius. Inoculum: dry mycelial spore transfer, liquid mycelial suspension, or lyophili. Period of incubation 48 to 72 hours to give pH ranking between 4.5 and 5.0. In the fermentation medium, sodium citrate 12.8 gram, sucrose 40.0 gram, ammonium sulfate 6.0 gram, magnesium sulfate heptahydrate 0.25 gram, mono potassium phosphate 0.15 gram, calcium carbonate 11 gram, manganese sulfate 0.01 gram, zinc sulfate 0.04 gram, and potassium dichromate 0.016 gram for the fermentation medium. In the optimization, volume of 50 ml culture per 250 ml in Erlenmeyer flask. Shaker in rotatory at 200 rpm. Temperature is 30 degrees Celsius. Inoculum per percentage 5% mycelium is transferred. Period of incubation is 4 to 6 days. The preparation of sample for assay: whole broth culture or acidified to pH 2.0 to 2.5 with 5N sulfuric acid and held for 1 hour before filtration through Wortmann number 4 filter paper. The paper disc method of assay with E. coli strain ATCC number 9637 is employed. Assay or run against a tetracycline standard. The tetracycline in crystallize. At the end of the fermentation, the culture broth is filtered to remove the mycelium. The filtrate is treated with N-butanol or methyloso-butylketone in acidic or alkaline condition for extracting the antibiotic. It is then absorbed to activated charcoal to remove other impurities. Tetracycline is eluted and crystallized. The uses of tetracycline. Tetracycline used to treat wide variety of infection, including asthma. It is an antibiotic that works by the stopping the growth of bacteria, venereal disease, atypical pneumonia, cholera, relapsing fever, rickettsial infection, and then plague. The storage for the tetracycline protection: keep at room temperature 68 and 77 degree F. Keep away from heat, moisture, and then light. Thank you.